Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For the section to discuss NCERT Class 12, the molecular basis of inheritance. Part 2, RNA. It is very the nucleotide residues has any addition of any OH group is present in the second position of the ribose. Very important. For example, when you take as in here in a it is in a ribose, it is a pentose sugar. Am I right? It is in a pentose sugar. So in the second carbon position, see here, it is a first carbon, it is in a second carbon. Am I right? In the second carbon position, is saying about as what? In the second position has in that OH group is present. Here OH group is present. That is called as what? Completely called as in a RNA. Is RNA also uracil is found in the place of that thiamine. That is in a 5-methyl uracil, another chemical name of the thiamine. So the DNA has any thymine is present, and then here uracil is present as any RNA. The instead of any thymine, the another name of the thymine is what? 5-methyl uracil. Okay, this is. Let us. The DNA as any acidic substance. The presence of the nucleus, the first to identify the Frederick Miescher in 1869. So already we know that in a first the nucleus first discovered by the scientist. The scientist was whom? Friedrich Miescher. And here he given as a name is what? That is nucleon. That is what? Nucleon. However, due as a chemical limitation of the isolation, such as in a long polymer intact as an illusion of the structure of DNA, it is remained completely elusive to very long period of time. So when to development of in this total DNA is time taking process. It is in a time taking process, but they're finally they identified as a long the DNA fragments to get find out after long time. Okay, it was only in 1953 that James Watson and Crick, they are based on the X-ray diffraction data proceed by as in the Murray Wilkinson and they are Rochelle and Franklin. These scientists, they are proposed as they are proposed a very simple but kind of famous double helical their model of the structure that is called as a DNA. So he is saying about as what? The first the nucleus discovered by as Frederick Miescher. Then the Frederick Miescher, when they discovered the DNA, the scientists James Watson and Crick, they are used as an X-ray diffraction method based on they are developed as a DNA. Then finally the scientist was whom? The Murray Wilkinson and the Rochelle and Franklin. In these two scientists, they proposed an X-ray diffraction based on be developed as a double helical structure of the DNA. They are completely to notice. Okay, let us here one of the hallmark of this proportion of was based the printing between as in two strands the polynucleotide chains. So here how many chains is present? There are two polynucleotide chains. However, the proposition was they are based on the observation that Erwin Chargaff law in double strands the based the Erwin Chargaff law that is in a double stranded DNA, the ratio between as an adenine and thymine, guanine and then cytosine, they are constant and equal to 1. So when this total DNA is present in this total DNA, the number of nitrogen bases as present, they are completely what? Like as an adenine, thymine, guanine, cytosine, they are equal 1. Means they are completely constant, they are equal proposition as what? That is in a 1 only. Okay, this is. Next, after the base pairing, the conifer is very unique. The base pairing, the conifers is very unique property of the polynucleotide chain. So when the pairing means that adenine to get bind with as a thymine, guanine bind with as a cytosine, they are completely here. For example, adenine to get bind with as what? Thymine, guanine bind with as what? Any completely cytosine. So this total pairing is they are uniquely they get arranged, and these are said to be as a complementary to each other. So when you take here this completely they form as a what? Complementary. Next, therefore as a sequence of the bases one strand is known and then the sequence of other strand can be predicted. So for example, in this we take as a two strands. Two strands. One strand information they can understand they can easily to get identified. How? Because here adenine here they have guanine, cytosine and adenine is present. What? Here they completely their opposite as what? Thymine is present, here cytosine is present, here guanine is present, here the thymine is present. So here they are already another strand you can easily to get predicted, means you can easily to get identified. Next, it is also is standard from as in a DNA, let you call as in the parental DNA. So for example, I took as in one DNA, the DNA is called as what? That is the parental DNA. That parental DNA they get act as what? Template for synthesis of any new strands. Very very important. So for example, here I am taking as what? This is in a parental DNA. It is what? It is in a parental DNA. In this parental strand, we call as what? Template. We call as what? That is in a template strand. And it is new that two strands, double stranded DNA. Let us we call as a daughter. This they produced as in a the identical to the parental DNA molecule. So let's see here. It is when we taking as an that parental DNA. In this parental DNA, they get producing as what? After few days, means after some time, it is to get replicates to get forms as in two strands. Means it is to develop as in a one strand, and it is also to get developed as in a another strand. Yes or no? So it is called as what is a template strand. It is in a completely their daughter strands. And then after because of this genetic material implication, the structure of the DNA is very clear. Because with how do they get formed? The genetic information means which base pair to get bind with as in a they are another bases they can have as in a clear information is there. So that purpose they get produced as in a clear cut of the DNA molecules. Okay. Next, what are the silent features of the DNA? Here, silent features of the double helical structure of the DNA they get followed as in a some under the futures. They are see one by one. First, they made up of two polynucleotide chains where the backbone is constituted by as the sugars, phosphate group and base pair projections inside. So when you take as any one particular DNA, they are followed as the three components as compulsory. One is in a nitrogen base, second one is in a pentose sugar, and third one is what? That is in a phosphate group. That is in a what? Phosphate group is must and should. Yes or no? Let us here. Second one is what? The two chains has an anti-parallel polarity. That means one chain has in a polarity 5 prime to as in a 3 prime, and another one is in a what is that? That is in a 3 prime to as in a 5 prime. So for example, here I took it as in a two strands. These two strands their polarity show as an anti-polarity. What? One is in a 3 prime to as in a 5 prime, and the one is what? 5 prime to as in a 3 prime. So they show as an anti-polarity condition. Let us. After the third one, once observe here, the bases of the two strands they are paired together hydrogen bonds. The hydrogen bonds are forming the bases. Adenine that forms within a two hydrogen bonds with as in a thymine. See here. Let us see here. The adenine to get bind with as in a thymine. How many bonds is present here? Two hydrogen bonds. And the opposite strand they are completely their vice versa. The similarly guanine they're binded with as in a cytosine. It has in a three hydrogen bonds is present. So here guanine to get bind with how many bonds is present here? 3 bonds. Cytosine or completely guanine to get bind with as in a 3 bonds will be there present. Okay. Next, after here always the purines comes opposite pyrimidines. Almost here purines comes as what? Pyrimidines comes, and this generates as in approximately the uniform the distance between as in a two strands of the helix. So when the helix will be to get forms, they are completely to get form as in a their anti-parallel region. Next, the two chains are coiled as in a right hand fusion. So when the two strands, for example, you take as in a here the two strands they get show as in a right hand fusion. Means the DNA to get forms as in a this manner only, right hand fusion only, and the pinch of the helix approximately 3.4 nanometers. So one pinch means this is one pinch. It approximately 3-4 nanometers, and it is here one more thing. 1 nanometer is equal to 1 billions of the meters. That is approximately 10 to the power of 9 meters, and it is they are wrongly up to as there are 10 base pairs of each turn. Each turn. So they given this is in a one completely one pinch. Here to here, it is what? One pinch approximately it has in a 3.4 nanometers. Okay, came the completely consequently the distance between as the two base pairs approximately how much? It is in a 0.34 nanometers. Say for example, when you take here distance between in these two, how much? This distance is 0.34 nanometers. Distance between the two base pairs, how much distance is there? Approximately in a 0.34 nanometers. And the plane one of the base the stalk over the another double helix. This addition of in a hydrogen bonds in a completely confers the stability the helical structure is takes place. So when the hydrogen bonds they get completely get bind here one by one, then what happen? It comes as in one stability. It comes as in what? One stability. Okay, this is. Next, compare here they given one question. Compare the structure of the purines and pyrimidines. Can you there find out why their distance between has in the two polynucleotide chain the DNA remain they are almost constant because they are saying about as what the completely the purines to get bind with as what pyrimidine. So that purpose it is the completely they are constant according to as an Erwin-Chargaff law. So adenine to get bind with as a thymine and guanine to get bind with as an cytosine. It is this completely they are constant according to as an Erwin-Chargaff law. Okay, next let us see the proportion of the double helical structure of the DNA. They are strictly in the explaining the genetic implication that became the evolutionary. So this total information they get explained which purpose it is mostly they are important for as any their genetic implications means generation to generations. It is very soon the Francis Crick the proposed as a central dogma molecular biology which is as an they start with as the state the genetic information follows as in a DNA to in a mRNA or in a as in a protein synthesis. Here, what is the central dogma molecular biology? Central dogma molecular biology means when the how do the replication is takes place. First, replication the DNA after the replication is takes place. Here, after the DNA replication is takes place, what happens? See, here this is a DNA replication, and the DNA replicates to get forms as an mRNA. It is this total process is called as a transcription. DNA is converted into as an mRNA. This process is called as what? Transcription process. And then this mRNA it is converted into as a protein synthesis. This is called as what? It is a part of the translation process. It is what? Completely translation process. Okay, this is called as what? In a central dogma molecular biology. Okay, it is a transcription process. See, in some organisms they show as a reverse transcription also. Some organisms here the given question, some viruses that follow information that reverse direction that RNA to DNA. Can you suggest a simple name of the process? See here what happened. So reverse transcription means or nothing but here completely in this mRNA. Here the mRNA to here what happened? Here takes place as any here that takes place as any the DNA synthesis takes place. It is a reverse transcription. Okay. After here the protein synthesis. Here protein synthesis. This is a reverse transcription when you observe as any retroviruses. When you see as any retroviruses. Okay, now this is in a completely information about as any DNA and then RNA. Okay. Remaining we'll discuss in next class. Thank you for watching.